Hello students, welcome to Vijetha IAS Academy, New Delhi, and myself is Lenin and I am the faculty of Geography and Science and Technology IAS Academy, and in this video we are going to discuss one topic from Science and Technology, especially from the biotechnology, because biotechnology is very important for Science and Tech from UPSC, both from prelims and mains perspective. So in this video we are going to discuss two small topics, both are interconnected. Okay, one is called as cloning, and the related topic we will discuss about the stem cells also, because stem cells is very important, especially from the mains perspective. Is it clear? So we'll see what is cloning, okay, and then we'll connect that one with the stem cells. Is it clear? So what is cloning? Okay, so cloning is nothing but we are having some replica of anything, means exact Xerox copy. Okay, so cloning is nothing but production of genetically identical copies, means exactly. Okay, so if we have seen some twins, right? So some twins will look like exactly replica of each other, but we cannot say those things as cloning. But here in this cloning, these two persons even will have their genetics also as same. Okay, exact copy will be there with respect to their genetics. Okay, it is the production of genetically identical copies of cells or organisms, and it is done through the method of asexual reproduction. Okay, asexual reproduction will be there in this one, and whatever the output that is coming, it will have exact replica. Okay, there won't be any difference in their genetical output. Is it clear? So such thing is called as cloning, but there are different techniques that are there which are involved in this cloning. Okay, so those two techniques are two types of cloning, which are reproductive cloning and second one is therapeutic cloning. Okay, so this therapeutic cloning is nothing but called as stem cell therapy. Okay, remember this therapeutic cloning is nothing but called as stem cell therapy. So we'll discuss what is stem cell and we'll see what is stem cell therapy, but we'll see what is reproductive cloning in the coming slide. So clear? So cloning is nothing but production of genetically identical copies of cells or organisms, means exact replica. Okay, print. Okay, Xerox copy will be there, and there are two techniques, one is reproductive cloning, second one is therapeutic cloning. Okay, so we'll see what is this one. So in reproductive cloning, okay, in reproductive cloning, okay, so what they will do is they will see, remember before that one, in human body there are only two types of cells. Okay, in human body there are two types of cells, one is somatic cells, okay, second one is germinal cells. Okay, see, these are nothing but egg and sperm. Okay, germinal cells are nothing but egg and sperm, which are responsible for forming babies, right? But other cells which are there in the body, rest of other cells, means for example skin cell, okay, or heart cell, liver cell, whatever the cell that are there, all the cells are called as somatic cells. Means apart from these two cells in the human body, okay, apart from these two cells in the human body, rest of all the cells are called as somatic cells. Okay, it can be any other cell, okay, that is called as somatic cell. So in this cloning, first what we will do, I want to make a copy of myself. For example, I want to make a copy of myself, right? So I will take one cell from my body, one somatic cell I will take, because in that cell, for example, this is the somatic cell I am taking, skin cell, I have taken my own skin cell. Okay, I have taken my own skin cell. What is there inside my nucleus? How many chromosomes will be there? 46 chromosomes. Who means these 46 chromosomes belongs to whom? Those 46 chromosomes belongs to me only, right? Right? So these 46 chromosomes of the somatic cell from the skin cell which I have taken from my own body is consisting of how many chromosomes? 46 chromosomes are there. Okay, so what I will do in this case, okay, I will take one ovum or egg. Okay, I will take one ovum or egg. How many chromosomes will be there in the ovum or egg? 23 chromosomes will be there, right? So in this ovum or egg, 23 chromosomes are there, right? What I will do is I will discard or remove this nucleus of the ovum or egg. Okay, so I will remove or discard the nucleus of the ovum or egg. So what is I am having? This is the ovum without any nucleus, right? Ovum will be there without any nucleus. but what I will do is I will transfer my 46 chromosomes. I will transfer my 46 chromosomes into this ovum. Okay, I will transfer all the 46 chromosomes here. Okay, 46 chromosomes are coming only from single person, means it is coming only from my body, right? And this is the ovum. So I will apply some electric power to this one, or some chemical reactions will be done on this ovum in the lab in such a way that it will start fusing, means it will start multiplying. Okay, so it will act as a zygote. Okay, this ovum itself will act as a zygote. So in the lab I will modify this ovum in such a way that, okay, by applying some chemical reactions or some electric current we will apply in this one in such a way that this ovum itself will start acting as a zygote. Once a zygote is there, see, if you see 46 chromosomes are there, okay, but all the 46 chromosomes are coming from whom? It's myself, right? But in normal cases, if I take this is the sperm and this is the egg. Sperm is having 23 and egg is having 23. So if these two are fused, then the zygote is formed. This zygote is having how many? 23 plus 23, 46 chromosomes forming, right? So 23 is coming from father and 23 is coming from mother. So a mix of father and mother characters will be there in this zygote, right? But in this case, what is happening? All these 46 chromosomes are taken from the single person cell, right? Means it is taken only from my skin cell. Means all the 46 chromosomes will be of my own genetic genes, right? So once these are transferred into this ovum, after discarding the nucleus of this ovum, and after this one in the lab we will apply some chemical reactions in such a way that this ovum itself will now act as a zygote. Okay, once it is acting as a zygote, means it will start multiplying, right? It will start multiplying. So what will happen? This zygote will start multiplying, and what it will form? It will form into a blastocyst. Okay, blastocyst is nothing but a cell mass. Means this single zygote will start multiplying into 2, 2 into 4, 4 to 8, 8 to 16, like that it will keep on multiplying like that. So a lump of cell mass, okay, is formed, which is called as blastocyst, and there is a prominence of this blastocyst. We will see what is it. Okay, inside, okay, this blastocyst will have different layers. Okay, in the inner layer of this blastocyst, okay, in the inner layer of the blastocyst there are cells. These cells are called as, okay, this is the blastocyst, and there are different layers of the blastocyst. In the inside or inner layers of the blastocyst there are some cells are present which are called as stem cells. Okay, remember there is one peculiar property regarding this one. Okay, because still, see, this is the single cell. This single cell has been multiplied and divided into a different group of cells, and it is formed into a lump of cell mass which is called as blastocyst, right? All these cells will have the same property, right? All these cells have same property because all these are formed from the same single zygote, right? And remember, till blastocyst there is no differentiation. Remember this term, no. Till blastocyst there is no differentiation in this cell mass. But after blastocyst, these whatever the different cells are there inside this blastocyst, right? Once some cells will convert into skin, some cell will convert into eye, some cells will convert into liver, some cells will convert into brain. Okay, means what I am saying, till blastocyst no differentiation, means all the cells look similar, but after blastocyst stage differentiation will start occurring. Okay, after blastocyst stage the same cells will acquire differentiation, means it will form into different specialized cells. Okay, all these are called as specialized cells. Why specialized cells? Skin has its own purpose. Okay, eye has its own purpose, liver has its own specialization, brain has its own purpose and specialization. So till blastocyst all cells are same. Okay, so it is having no differentiation till blastocyst, but after blastocyst stage these cells whatever that are there inside the blastocyst will start multiplying and differentiation will start occurring, so that these cells will start forming into different type of organs within the human body. So this blastocyst is very important for us, because this the inner layers of the blastocyst, whatever the cells are present, these are called as stem cells. Is it clear? So once this blastocyst is formed, remember, see, I am erasing this one. Okay, so this, till this blastocyst is formed, if this blastocyst keep on multiplying like this, and a stage will come, a kind of fetus will develop, and this fetus will finally lead to child birth, right? So this fetus will lead to the childbirth. Means once I am transferred this one, this blastocyst will be formed here, this zygote will convert into blastocyst. Means this zygote I will transfer this one into mother's womb. Okay, in the mother's womb or in the lab also I can do this one. So either it will convert into blastocyst, after the blastocyst it will convert into fetus, this fetus finally will result into the childbirth, right? Whatever the child that has been born, okay, whatever the child that has been born, it is having whose DNA? All these 46 chromosomes, right? The same 46 chromosomes are there here also. So if I take any cell of this child, any cell of this child is having the same chromosome, means they genetically, these both, this skin cell and also the child which is born from this one, genetically both are similar. So whatever the child that will grow with time, it will look almost similar. But intelligence, all these things will be depend on environment also, because genetics and environment will play a major factor in intelligence. Okay, but if you see genetically, these two persons will be having same genes, because I have taken all the chromosomes from the single person, and these chromosomes after inserting into the ovum, this will be made or formed into a kind of zygote, blastocyst, fetus, and finally childbirth, right? So this entire process is called as reproductive cloning, because I am reproducing a child here, right? As I am reproducing a child here, it is called as reproductive cloning. Is it clear? But remember, this is reproductive cloning, right? So the second technique which we have seen is therapeutic cloning, right? Therapeutic cloning. In this therapeutic cloning, what we will do is, okay, whatever the zygote that has been formed here, the same technique I will use in this one also. Whatever the zygote that has been formed, this zygote will be multiplied so that it will convert into blastocyst. But what is there inside this blastocyst? What are these cells? These cells are called as stem cells, right? Stem cells. In this case, I will not insert this zygote into the ovum, zygote into the mother's womb. Okay, instead of that one, I will try to multiply this zygote in the lab itself so that it will form into blastocyst. From this blastocyst, okay, from this blastocyst I will extract these stem cells. Okay, from this blastocyst I will extract these stem cells. Okay, so in previous case, blastocyst is converted into fetus, fetus will be formed into a child, so that finally we will have a child, baby will be born, right? But in this case, baby is not borning. Instead of that one, I am stopping everything at this blastocyst stage itself, where I am extracting the cells, the inner cells of this blastocyst, which is consisting of these stem cells. And we have seen what are the properties of these cells, whatever that are there in the stem cells, they are not differentiated, means they are not specialized yet, but after blastocyst stage they are specialized, right? So I am taking some cells from this blastocyst which are not specialized. Now I will use these cells in converting these cells into different purpose, because the same zygote has been formed into blastocyst. From this blastocyst, whatever the cells are there, some cells are forming into liver, some cells are forming into blood, some cells are forming into heart, some cells are forming into lungs, or different organs are developed from this same cell, right? Means what it is indicating, these cells have the capacity, to convert into different organs in the human body. So if I extract these stem cells, okay, if I extract these stem cells and someone is having heart failure, okay, if some person is having heart failure and if he is not getting any heart donor, what will be the case? Within few days he will die, right? So to avoid that one, okay, to avoid such things, what we are doing is, I will take a cell body, okay, from a cell from his body I will take, okay, I will make this zygote, so that I will try to make the blastocyst, and I will take the stem cells from that one, and from that stem cells I will try to build a heart of his own in the laboratory. Okay, remember, I will try to build a heart of that defected person whose heart has been failed. I will make heart of that own person from his own stem cells, so that I will once again reinsert that into his heart, into his body, so that once again it will start functioning, so that we can save the person. Okay, that is nothing but we are nothing but giving a kind of therapy, right? That's why it is also called as therapeutic cloning, or it is also called as stem cell therapy, because we are using stem cells and finally we are treating that heart failure person, right? So that's why it is also called as stem cell therapy, or it is also called as therapeutic cloning, because I'm making the clone of its cell, the clone, right? I'm not making this one to form as a child. Instead of that one, I'm using this stem cell, so that I'm using this stem cell for different purpose, like making a heart or making a liver or making some other organ of the body, so that the defective person can be treated using these things. Okay, this is nothing but called as therapeutic cloning or stem cell therapy. But we'll see what is stem cell, all these things also. I hope you have understood the cloning. Cloning is nothing but making the genetically exact replica or copy of a person is called as cloning. And there are two things in this cloning. One is reproductive cloning, where I will take the somatic cell, and as we are transferring, okay, remember, as we are transferring this nucleus or this entire nucleus into this one from the somatic cell, it is also called as somatic cell nuclear transfer technique. Remember, this question has been asked by UPSC multiple times. What is somatic cell nuclear transfer technique? Because I am taking somatic cell, from that somatic cell I am taking the nucleus into the discarded ovums. Okay, so whatever the ovum is there, I am discarding the nucleus of the ovum, and I am transferring the nucleus of the somatic cell into the ovum. That's why it is called as somatic cell nuclear transfer technique. And using this one, I am making blastocyst. From this blastocyst, I am extracting stem cells. Okay, so in reproductive cloning, I will go for child, so that it will born, and in this reproductive stem cell therapy or therapeutic cloning, instead of going for child, I will extract the stem cell from the blastocyst itself, so that I can use this stem cell for different purposes, like treating any blood cancers I can use, or I can use for making some bloods also. Some blood is there, okay, which is very rare bloods are there. Okay, so for example, Bombay blood group is there, very rare. So I can make that rare bloods also in the lab, or I can make O negative bloods, which can be used by different purposes or different persons. Like that, I can make different bloods, organs, different organs. I can make some organs and I can use those organs for testing also. Till now, clinical trials we are doing on small animals, right? Rats, all these things we are using in this clinical trials, and we are killing a kind of, means we are creating a kind of pain, because we are using these drugs, or we are using those drugs and testing on those animals, right? Now what I will do, I will make some organ and I will test my medicine on this one. Okay, so like that, I can make clinical trials in the lab itself by making some organs from here. Okay, I can use for organ transplantation also. I can use for treating of some cancers, very dangerous cancers also I can treat using this stem cell therapy. And recently some people are even being treated who are having HIV also. Around 5 people have been treated using this stem cell therapy, and they are cured from HIV disease also using the same stem cell technique. So we will see briefly what is stem cells and what are the different types of stem cell, and we will wind up the class, right? Is it clear? I hope you have understood the cloning topic. Okay. Okay, so stem cells. Okay, if you see zygote, blastocyst, and finally it will lead to the forming of some baby and adult, right? So based on, see, there are different types of stem cells. Okay, so there are different types, means there are certain properties of the stem cells. First we'll see the properties of the stem cells, and then we'll see what are the different types of stem cells. See, properties of the stem cells is nothing but, sometimes if you extract any stem cells and if we store these stem cells in a particular conditions, for example, some conditions like -196 degrees centigrade, if I extract the stem cells and if I store these stem cells in -190 degrees centigrade temperature, and in future after 15 years, 20 years, if I stem cell and if I remove from this temperature and if I start using that one, then these stem cells will start once again functioning. Okay, so such a property of stem cell is called as self-renewal. Means even conditions, if I remove all those conditions, then once again we can retrieve back and we can use such stem cells. Such property is called as self-renewal. And second property of the stem cells is, what is that? Unspecialized, which I have discussed, right? Means unspecialized is nothing but these stem cells can form into any other type of cells in the body. Okay, that property is called unspecialized property of any stem cell, because after blastocyst stage, each and every cell will start becoming specialized cells. Means each and every cell will start either forming into eye or liver or heart or brain or into different organs. That's why stem cells are unspecialized. Another property is totipotency. Totipotency is the property of the stem cells, where that stem cell, totipotent stem cells are those stem cells which can be formed into any organ in the human body. Means if I use this totipotent stem cell, it can form into any organ. I can make, using this totipotent stem cell, I can make any organ in the human body, right? And pluripotent stem cells, if I use, I can make more, means different types of organs, but not every organ. For example, I can make any organs except placenta. Except placenta, I can make other different organs or other different types of cells in the body. Okay, in this case, even we can make placenta also using totipotent stem cell, but using pluripotent stem cells, we cannot make all the types of cells, but we can make many types of cells except placenta. If I take multipotent, we can make types of different organs or cells in the body. And unipotent means if I take skin cell, I can make only skin cell. If I take liver cell, I can make only liver cell. Like that, only one type of cell can be made from this unipotent stem cell. Okay, these are some of the properties of the stem cell, like self-renewal, unspecialized cells, totipotency, pluripotency, multipotency and unipotency. Is it clear? Now we will see what are the different types of stem cells. Okay, so based on, okay, types of stem cells based on the source, means from where we are extracting, based on that we are having different types of stem cells. Like if I are taking the stem cell from the embryos, okay, embryos, these are called as embryonic stem cells. If I am taking the stem cell from adult human beings, for us, like any person, okay, if they are taking some stem cell from my body, then it is called as adult stem cells. If they are taking from the fetal, okay, that is called as fetal stem cells. If they are taking from infants, perinatal stem cell. Perinatal can be of different types, like amniotic. Okay, so what is the placenta is there, right? Inside that one baby is there in the womb. Surrounding that baby, one fluid will be there called as amniotic fluid. Okay, so that will also have some sort of stem cells. Okay, if I extract stem cell from that one, it is called as amniotic stem cells. Okay, and after the childbirth, okay, whatever the umbilical cord that is connecting the baby and the mother, okay, one cord will be there, right, which is connecting the mother and the baby. So after birth, they will cut that umbilical cord, right? That umbilical cord will also have certain blood. Okay, that blood will have stem cells, which is called as umbilical cord blood stem cells, right? And these days it has becoming very popular that every doctor is asking to store this cord blood, because in future if your child is having any issue, health issue, we can use this stem cell, we can use these stem cells and we can treat the babies like that. They are telling, so that because of that one, this cord blood stem cells storage has become very popular in the recent times, right? So these are based on the source from where they are extracting, and based on the method how they are derived. Based on method means how they are derived, there are different types, like IPSC, induced pluripotent stem cells. I'll discuss what is induced pluripotent stem cells, but remember IPS, induced pluripotent stem cells. And second one is somatic cell nuclear transfer. Just now we have discussed, right? Somatic cell nuclear transfer, the technique is used so that zygote will be formed, blastocyst will be formed. From that blastocyst, I will extract the stem cells, which are called as somatic cell nuclear transfer. Another one is neural stem cells. These neural stem cells will be used for the treatment of any neurological problems. And hemophotic stem cells, these are the stem cells which are allowed in India, and using this one we are treating some kind of blood cancers in India. Okay, remember we are using this type of stem cell for treating blood cancers. Okay, blood cancers are treated using these stem cells in India. Okay, so but we will see, we have discussed somatic cell nuclear transfer, right? We will see what is induced pluripotent stem cells, right? So here what it is, the term pluripotent stem cells we have discussed, right? Pluripotent means they can form into multiple types of organs or cells in the body except placenta, right? So pluripotent stem cells are those stem cells which cannot form into placenta, but it can form into different other types of organs or cells in the body. But what I am doing, inducing, means I am inserting something forcefully. I am inserting something forcefully so that that cell is acting as a pluripotent stem cell. Means I am taking one cell. Remember, I am taking a cell. Okay, I am taking a normal cell. This normal cell is induced or forced in such a way that this normal cell is now acting as pluripotent stem cell. This normal cell is now acting as a okay, so I'm inducing something into this normal cell in such a way that this normal cell is now functioning as a pluripotent stem cell. Okay, so how it has done is, the scientists have observed that there are 4 cells in this cell. There are 4 genes. Okay, in this cell there are 4 genes, okay, which are called as Yamanaka genes. Okay, because it is the scientists who have found this one. So these 4 genes they have found in the cells in the human body which are responsible for the specialization. Okay, so these 4 genes are responsible for the specialization of any cell in the human body. So what they have done is, they are modified, means scientists have modified these 4 genes in such a way that once again this normal cell which has become specialized because of these 4 genes, because of the modification of these 4 genes, this normal cell once again is acting as a pluripotent stem cell. Remember, these 4 genes are identified by a person called Yamanaka. Okay, so based on his name, these genes are named as Yamanaka genes. For this discovery, even that scientist has got even Nobel Prize also in the medicine. Okay, remember, even that scientist has got Nobel Prize for the medicine for the discovery of these 4 genes which are called as Yamanaka genes, which are reduced in such a way that when we modify these 4 genes, then any normal cell in the human body can be transformed into a pluripotent stem cell. Means in this case, see, previously we are a kind of killing that blastocyst, right? Because once we extract any stem cell from the blastocyst, then there won't be any further continuation of that step. Means there won't be any forming of baby, because there won't be any fetus. Once fetus is not there, there won't be any baby, right? Means we are killing a person, right? After zygote, we are having blastocyst and we are extracting this stem cell from that one, and indirectly we are killing that person, because we are not giving any chance to giving birth of that blastocyst, right? So we are killing that one person, right? So a lot of ethical concerns have came here. But now if I see in this case, I am taking any skin cell, okay, I am taking a skin cell from my body, and now in that skin cell I am inducing some changes in these 4 genes of that skin cell, so that this skin cell, after inducing some changes in those 4 genes, the skin cell now it is acting as a stem cell, which is nothing but a kind of pluripotent stem cell. Okay, so now is there any chance of giving any, means killing any zygote or killing any fetus or embryo? No, right? So because of that one, as there is no ethical concern, this method has becoming more popular, and because of the discovery of this technique, okay, these scientists have been awarded Nobel Prize also. Is it clear? So this is induced, because we are inducing something into normal cell so that the normal cell is as a stem cell. This is induced pluripotent stem cell, somatic cell nuclear transfer, neural stem cells, and hemophytic. India has allowed for treating different types of cancers in India. Is it clear? And these are some of the applications, okay, which we have already discussed. Using stem cells, we can make muscle cells, okay, we can make intestinal cells, we can make liver cells, we can make heart cells, we can make nerve cells, we can make blood cells also. Apart from those things, there are some other benefits also we can use for regenerative medicine, which is nothing but therapeutic cloning. Okay, stem cell therapy is nothing but called as regenerative medicine. Stem cell banking, artificial bloods we can make, we can test different drugs also, we can make different organs also, organ transfer also using these stem cells. Is it clear? So stem cell therapy, the same which we have in stem cell therapy, that once after that required position, they will try to treat or cure the disease, or otherwise we will try to make some organs also. We will insert those organs into the human body so that the damaged organs will be replaced with the healthy functional organs, so that organ transplantation also can be done using this stem cell therapy. Is it clear? So these are the small topic which I want to discuss. In this small video, I hope you have understood and it is clear. Thank you.